Well, good afternoon, everyone. Welcome to the final neuroprosthesis seminar for the calendar year. I'll introduce our speaker in a second. We have a continuing great lineup into the new year, so pay attention to the announcements. Let's see if I can remember who they are. So January, we're going to be doing strategic planning, so we won't have a seminar. Andre Machado, Emery Brown. Who? Oh, anyway, it doesn't matter. We'll figure it out. We have a very great lineup for our neuroprosthesis speakers coming up through the end of the spring.
It's good to know your memory is going too.
No, I can remember one person, but I can't remember their name. That's terrible. That's even worse, maybe. Doug Weber is one, and Vivian Mouchouar. Yep. Okay, so you're in good company. You're in good company. Well, anyway. I'll stop while I have a little shred of credibility left. So it's really my pleasure to introduce our speaker today, Lee Miller. Lee has a PhD from Northwestern University in physiology. He did a postdoc in the Department of Biophysics and Medical Physics at the University of Nijmegen in the Netherlands. And he's on the faculty in the Department of Physiology at Northwestern, where he's currently the Edward C. Stuntz Distinguished Professor of Neuroscience. He's the president of the Society for the Neural Control of Movement. He's a fellow of the American Institute of Medical and Biological Engineering. Lee is an old friend of mine. We went to graduate school together long ago. We still remember that far back, and we've continued to maintain contact and talk about getting research projects going all the time. So, yes, I'm very happy to have Lee here, and as you can see his title, In Pursuit of a Biomimetic IBCI for Dexterous Hand Use Following Spinal Cord Injury. So, Lee.
Thank you, Bob. I just wanted to shred a little more of that famousness of Bob. The one thing that I remember from our master's degree that we did together was we had a class project in some class, I forget, in which we had to make a double-sided circuit board to do something. I can't remember what it did, but we managed to mirror the two sides so that they didn't line up properly. That's my memory.
It was Lee's fault.
That's back in the day when you actually used tape on paper, to do the thing. Anyway, it's good to be here. I don't quite remember when the last time was, but in addition to my connection to Bob, I'm connected to a number of other folks here through either formal or informal collaborations that really have been critical to my having gotten my career going. And I'm going to tell you a little bit about that story, which is something of an arc from early work with monkeys through our current work with some computational methods that we're applying to those data, and then finally translating this to humans. And this is partially also campaign call here to see if I can motivate a few people to join me in this project, which can only be done properly here at CASE in the FES Center. So outlining the basic talk is I want to start by demonstrating this biomimetic BCI that we did now a bit over 10 years ago in several monkeys to restore grasp. And then trying to extend that BCI from the very simple paradigm that we use to the lab to something a little bit more like what your activities of daily living would need to be like. And then finally, as I suggested, ways in which we'll begin to translate this to use in paralyzed humans. And I should also say along the way, if there are questions that come up, please feel free to stop me. Let's keep this informal and have a good conversation along the way. So what's a BCI? This is a BCI. This is also 10 years ago, and this comes from the University of Pittsburgh, of which I have now become a part of an extended consortium known as Cortical Bionics that includes the University of Chicago and now Northwestern. And this was a woman with tetraplegia with 2 Utah arrays implanted in her head, controlling 7 degrees of freedom of this robot, which is the 6 endpoint degrees of position and orientation and opening and closing. And you see she's doing quite well. However, we didn't evolve over millions of years to control the speed of rotation of the joints of a robot. What we learned to do was to contract muscles and stiffen joints if we needed to, and push on things and so forth. So that's the nature of the BCI that I have been working to develop. And this is another video clip borrowed from you folks. I'm bringing lots of coals to Newcastle in the course of doing this experiment. But this is one of the participants here in the FES Center with these same kinds of implanted arrays. And instead of using servo motors and robots, he is using his own muscles, which are being made to contract by stimulation of perhaps a combination of intramuscular muscles, electrodes, and nerve cuffs. I don't know what the particular combination implanted here was, but the way in which this control was effected was really close to what it was in that first video that I showed you. They decoded kinematic parameters of the movement and used the muscles instead of servo motors to make those kinematics happen. So it's still, in some sense, as though there's no mass involved, because it's a kinematic control signal decoded from the motor cortex that the participant learns to make happen by contracting their muscles through this FES. So what I'd like to do is sort of give back the direct control of muscles to a person like this, rather than going through this intermediate kinematic phase. This is probably not terribly necessary, but this is the Utah array, which was in both of those participants. It's in all of my monkeys. It's an array of 100, or in some cases 128 electrodes, each of which records neural potentials, as are shown here. And this just shows an implantation of two of these devices in one of my monkeys, one in motor cortex, one in somatosensory cortex. And I don't think I need to say more than that unless someone doesn't know what I'm talking about. But what it allows us and the folks here working with their participant to do is to record from roughly 100 neurons at the same time. And that's the color-coded rasters at the top here. Is there any chance of getting these lights down a little bit? If so, that'd be great. So in this case, we had about 70 neurons, and you can see that there are bursts of color here that correspond to the firing. Thank you. Perfect. Don't fall asleep, though. The bursts of firing of these neurons, and at the same time, I should have pointed out here, this is what the monkey was doing. He's reaching out, grabbing this ball, dropping in the tube at the bottom, and he just, like Sisyphus, keeps doing it over and over. These are the bursts as he grasped that ball 3 times, and these are the patterns of muscle activity from 3 different muscles of the wrist and forearm flexors that are involved in producing this behavior. So as you can see, the repeated grasps have fairly consistent patterns of muscle activity, although not identical, and they vary across muscles, of course, because the different muscles have different functions. And what we did then is essentially what others do when computing a kinematic decoder. We simply did the essence of multiple linear regression. For each of these neurons that we recorded from, we looked at their firing rate in 10 bins back in time. So 10 times 70, roughly, regressors were used to fit the activity of each of these muscles. So that you can think of it as a multiple input impulse response, a Wiener filter. There are many names for the same thing, but it's a simple linear mapping between cortical activity and muscle activity, which we can then test in this monkey, because we can also record the EMG, as I said. So, sorry, I already showed you the recorded EMG. The blue now is the cross-validated prediction of that EMG. And you can see how well the blue overlies the red. That's the point here. We can look at the brain, and more or less, as other folks decode the velocity of hand movement, we can predict the patterns of activity of these 3 different muscles. And we did more than these 3, but these are just 3 examples. And you can see the quality of the prediction ranges from an R squared of about 0.5 up to 0.8, which was typical of the kinds of predictions that we could get. Okay? So this gives us the means to control FES directly from the motor intent of the motor cortex, rather than without having to go through this kinematic model. Or at the last moment, I took out a slide about the freehand prosthesis, which was developed here by Hunter Peckham and many others. I'm going to assume most of you are familiar with it, but basically it uses the residual 1 degree of freedom that a person with an injury at C5 might have remaining. And that 1 degree of freedom control signal is used to control the stimulation of 8 12 different muscles, which all has to be pre-programmed to cause them to contract at the right time. Here what I'm doing is giving all that control back to the monkey so that he can use his motor cortex to effect that same kind of control over implanted electrodes. So this is the experiment that a student, Emily, and a postdoc, Christian, did in my lab a number of years ago. And I've told you about most of the steps of it. We basically make these real-time predictions. We map those onto the pulse width of the stimulator, stimulate muscles, which we have paralyzed by doing a peripheral nerve block. And that's the sort of key here. This is our model of a transient paralysis. So it has many of the aspects of a spinal cord injury and doesn't have many of them. Most importantly, we can turn it on and off. So that following the nerve block, this paralysis persists for a couple of hours, and then the monkey returns to normal. So that means we can record normal signals from this monkey, computer decoder, and then test it. And that's what this next video shows. So this is a monkey with the peripheral nerve block, but before we've turned on the cortically controlled FES. And he reaches out, he tries to grab the ball, but he can't use his hand because of the nerve block. But presumably the hand area of the motor cortex is still doing more or less what it would normally do. So if we could take those signals and get them to the muscles, he might be able to use his hand. And that's what he's doing here. So now we've taken the pattern of activity in the motor cortex, figured out what he was trying to do in terms of muscle activations, and electrically stimulated those muscles to cause them to contract in a voluntarily controlled manner. So he's coordinating that distal electrically driven muscle activity with the proximal completely normal shoulder activity in what looks like a pretty natural sort of manner. Okay? So that's what it looks like on video. This we had him doing a power grasp task as well, in which we'd actually measure the force that he was producing. And in fact, he had to produce a particular amount of force in order to get into these different force targets at different levels. So this is, more or less the screen that was scrolling back and forth in front of him. And this is the force that he produced in order to get into the target and get the reward. Except for this catch trial right here. We wanted to make sure that this was really, really working and that he wasn't somehow using residual normal control to accomplish the behavior. So we turned off the prediction and the stimulation part of this pipeline. So you can see in red, we were predicting this amount of EMG in these two different muscles, but we didn't stimulate. And so he was unable to generate any grasp force at all. And so what's interesting is he squeezed once, it didn't work. He tried again, didn't work. And then we turned it back on and he was able to complete the next trial successfully. So he really does have this voluntary control over this grasp, at least the timing of the grasp here. The next slide I really quite like because it shows his ability to control. Well, I showed you he controlled the timing and the force as well, but here's another aspect of what he's able to do with this FES, which I find rather nice. Up here at the top is our two graphs of the nerve block effectiveness, which, as I said, lasts a couple of hours. When we try to make sure that we only do the experiment during this period when it's fully effective. This is sort of the dark side of FES. When you stimulate muscles or nerves electrically without going through a synapse, it reverses the order, the normal recruitment order, and you end up fatiguing muscle much faster than it does under normal activation. And that's what you see here. Within 30 minutes, the strength that he can produce drops down to 30 or 40% of normal. And it's a significant issue, especially for big, heavy limbs. Using the hand, perhaps not quite as much unless you need to generate a lot of force. And that's what we were asking him to do. The highest force level was fairly substantial. I forget what percentage of max, but significant. But early and late in this session, which as you can see lasts almost 2 hours, he was able to get the force target up into the highest level target. What's interesting about this part of the story is early on, this is the pulse width for one of the muscles, I don't remember which, that we predicted and used as the basis for stimulation in order to get into that upper target. Toward the end of the session, he needed 3 times the muscle activity to get that same force because his muscles had fatigued. But he went ahead and did that, just as you would do if your muscles became fatigued by, lots of exercise. You'd have to try harder. He was trying harder here, which he could do by virtue of the predicted EMGs. Okay, so that's the basic demonstration for you. And then what I want to do now is show you how we're working to extend that in several different ways to what could be a more useful kind of prosthesis. So part of what we're doing here is recording wirelessly, both from the brain and from the muscles, while the monkey is in a fairly large home cage, a couple meters wide and meter high and back to front. So here he is hanging out in the cage. You can't see terribly well, but there are bars across here, and you'll see what he does with them. In addition to scratching himself occasionally, he'll reach up and take a treat. And I should point out while it's scrolling here, this is neural activity, this is muscle activity. He just walked across these bars, grabbing him with a power grasp, and now he goes down to an instrumented power grasp thing. So there's a variety of natural sorts of things he can do spontaneously. He does this locomotion, walking back and forth in the cage. He takes treats, he does the power grasp. And we're interested in looking at this range of different sorts of behaviors. It's not shown here, but we also had enrichment games that we'd give the monkey. So one example is a cardboard tube full of scraps of paper and nuts or something like that. And you put it in, he tears it apart in about 15 seconds and finds the nuts. And I'm showing people who were involved in this along the way if I don't remember to mention them each time. But Schwen is a postdoc currently in my lab who's done a lot of the recent analysis of the data from the cage. And in fact, he just finished his final collection of data from this cage, some of which I'm telling you about here. So we also had video from the cage, and we were interested in trying to get it analyzed automatically with some sort of deep network, but it didn't work so terribly well. So Schwen went in and the hand identified periods of particular kinds of behaviors. And the three that I'm going to highlight here, and that he did a bunch of analysis with, was the power grasp here on this device, the power grasp that he used while walking across the cage, and a precision grasp, which he did just to take treats from the experimenter. And on the right, you can see the neural activity at the top, the EMG at the bottom, and you can see that there are patterns in time across these neurons which differ from task to task. And he tried to align all this up to the onset of the grasp. This is 2 additional trials for each of those, and you can see that there's a pattern that recurs for each of these identified behaviors. But obviously, there's variation here as well. So the first question is, how well, I showed you prediction, a few EMGs that we predicted during this ball grasp task. Here's a bunch more, and they're really impressive predictions, if I do say so myself, although it's Schwen, of course, who did them. But within the power grasp task here, he gets very nice predictions, the perch bar grasp and the precision grasp. But he's training and testing on the same behavior. What happens when he tries to extrapolate from one to the other? Dustin's nodding his head. He knows what happens. It falls apart. So in particular, the one that you think might work, going from the power grasp to the power grasp on the device, just doesn't work at all. Probably because in order to walk, he's coordinating all 4 limbs instead of sitting here and using one. The proprioceptive input that's coming is completely different. The tactile input is different. So it's a very different context for this behavior. And the prediction doesn't work at all. A little better between power grasp and precision grasp, but if you look at a summary kind of diagram, the diagonal is the within task predictions, and the off diagonals, if they're above 0, they're not much above. So basically, it doesn't generalize.
What if you train them all together?
If you train them all together, it works better. And I'm going to have a slide with that coming up in a moment. Yeah, good question. Extrapolation is very difficult, so you can basically not hope that it'll work. There is a linear model that works for all of these, but not as well as the individual ones do. And that may give you a clue to one of the things that Schwen is trying to do, in fact. But before I get there, I want to take a bit of a diversion and talk about the concept of a neural state space. I understand there's people out in the cloud somewhere, so you can't raise your hands. But I'm curious, how comfortable or how familiar are most of the people here with this concept of a neural state space? Do you either already know or can you guess what I'm talking about? Okay, not so much, few. But basically, this illustrates it right here. All you do is construct some n-dimensional space, each axis, each dimension of which is the fire rate of a single neuron. So within this three-dimensional space, you can locate the state of the brain at any particular time with a point in that space. And I'll just scroll through, and this comes from Cunningham and Yu, quite nice theoretical paper about these kinds of ideas a few years back. And eventually you can trace out this pattern of state space positions to form a trajectory. And it turns out, even this is a cartoon, of course, but within this cartoon of three space, all these points lie along a single two-dimensional plane. And that turns out to be the case, although it's a larger space to begin with than three dimensions, and the plane is a hyperplane of some sort. But you can think about extending this three-dimensional example to the 100 dimensions that I can record with an array in the monkey's brain and ask what the dimension of that might be. And it turns out it's on the order of 10 or something like that. So it's a small manifold or subspace that lives within this larger embedding space of the neurons that we record from. Everybody good with that idea? Yeah. You can't be sure. Well, you could go in and try to measure it, and that's one thing that we have done. And a bunch of people are interested in this. And you can do PCA and try to find the knee and say, it's 4 dimensions, or you can use nonlinear methods. We have a paper a year or so back with simulated signals trying to develop some principled ways of answering your question. And we did that, and it's about that order. But there's an additional concern that maybe it's because we're doing simple stuff, and that's why the dimension looks so small. And so that was part of our motivation for using this cage. I'm not planning to show it to you, but the dimension, if you look at everything in the cage at once, goes from 6 to 10 or so. So it goes up, but it's still on the order of 10. And that's an interesting question, in fact. Why is it that? And what might we be able to do to make it higher than that? Was there another question? Yeah.
I was going to ask what you just said is the dimensionality related to the complexity of the task?
Yeah. I mean, in some sense it has to be, but the real question is, until we can get a monkey playing Chopin, is it ever going to get above 10? Okay. What's that?
That might point nature.
We've not started on it yet. Okay, so there's the time trajectory. So that was a cartoon, of course. This is actual data from Schwenn's data from the cage. So this is starting from that 100-dimensional space, and I think he used PCA here. I'm not quite sure. He's used both PCA and some nonlinear methods to do this kind of thing. But he's reduced this 100-dimensional space now to 3, on which he's going to show us the trajectories that these different kinds of behaviors take through this space. And we'll rotate it, and so you can see that the wrist movement, the power precision, power grasping, crawling, and so forth, live in different parts of this 3-dimensional space. So it's not terribly surprising that our ability to extrapolate from one to the other isn't going to work if we just don't have training data in the relevant parts of the space. So this, in some sense, gives you something of the idea of what we're up against as we try to move to these more complicated spaces. And it also introduces the idea of these latent signals. I've not used that term yet, but as the state space traces out a trajectory when the monkey does something, you can think about that as another signal, just like the firing rate of a single neuron. And so a lot of people, ourselves included, have been interested in what information there may be within this set of roughly 10 latent signals that underlies the monkey's behavior. Sorry, here's the answer to your question, Bob. So he's tried transformers and some fancy neural networks and so forth, and some other folks in my lab have as well. But the straightforward approach is to recognize the success at single behaviors, recognize the fact that they live in different parts of the state space, and just do a piecewise linear decoding scheme. So this is an entirely unsupervised approach. He uses either PCA or local linear embedding, other nonlinear methods, to reduce the dimensionality, and then k-means or other methods for finding clusters. within this space, and then simply computes a decoder for each of those clusters and applies it whenever he finds a fire rate in a different part of the space. So this is an example of data recorded continuously from the cage with the monkey doing a variety of things. And ordinary decoder Bob means a decoder trained on everything. And it does reasonably well, but you get at least a 20% increase or so by going to this piecewise linear approach. So we're continuing to look at different ways in which we might be able to get decoders that span this broader range of behaviors more accurately as a control source. All right. Oh, so this just shows you how the R-squared increases with numbers of clusters, and surprisingly, using PCA, he did better than with LLE. We don't have a good answer for that. We both expected the other way around, but it turns out they said the clusters each of these finds are functionally somewhat different. And for whatever reason, the PCA-based one has worked better so far. And here is the linear one. Okay, so that's one example of ways to move to a more diverse set of behaviors and apply these same kinds of methods to them. So now I want to talk about some of our efforts to use these latent signals in order to produce decoders that may work in humans, EMG decoders in humans. So this was the first paper with a Utah array implanted in a human to do brain control, back from 2006. And there had been some monkey work prior that developed this method that they exploited, but I think this was probably the point at which the term observation decoder was first used, or at least certainly became popular. The problem, of course, is unlike with my monkey, they can't record the output signal from a person who can't move their arms. They can record from the brain, but they can't record EMGs or the movement of the hand. So instead, they have the person watch a trajectory that is drawn out on a screen in front of them, and the term observation is a misnomer. They're asked to attempt to make that movement. And to the extent that they're able to modulate the neurons that would have caused that movement, now they can go and record a decoder between the brain activity they record and the trajectory that they ask them to make. And that works quite well. Typically when it's done, it's an iterative kind of process where you come up with a decoder, you test it and get some more data, refine it, and so forth. But that's the approach to building a kinematic decoder these days. The problem is, I can't show someone a pattern of muscle activity and ask, try to make this pattern of muscle activity. Maybe flexing and extending the elbow would work, but certainly not dexterous hand movement. So we need another approach. So now I'm going to tell you an unrelated story just to convince you that these latent signals are real and they contain information that is stable and potentially useful to us as well. So one of the problems that we all have, whether we're working with monkeys or humans with these chronically implanted arrays, is the neurons that they record change over time. Of course, over the course of years, you typically completely lose neurons as well. But before that happens, the identification of the neurons just changes. So if we build, and you can see an illustration of that here, the different wave shapes that are evident just within a few weeks. And if you build a decoder back here at the beginning on day one and continue to apply that fixed decoder over time, in this case it was reaching movements from one of our monkeys, it stops working, in this case about a month out or so. It is not the case that the neurons all went away, because if you compute a new decoder each day, it's still there. The information is still there. But this means you need to compute a new decoder every day, and the person needs to learn to use it every day and so forth. So that's not desirable. And we're interested theoretically whether there's a better approach to this problem as well. So Matt, Juan, and Raid went together on this rather nice story that brought together somatosensory data as well as motor, but this is data from motor cortex during that reaching movement that I showed you. So here is another set of these latent trajectories. Whoops. And they're color-coded to correspond to the direction of the reach. So you can see they're basically parallel arcs through different parts of this neural state space, nicely ordered for the direction of the reach. That's back on day one. Day 32, they look like this. So we're using the same projection to find these latent signals on day 32 that they did on day 1, and it doesn't work anymore. It's just complete spaghetti because, or possibly, the hypothesis is, because these latent signals are embedded within a different high-order embedding space. We're recording different neurons. But if there is something like a real latent representation of the monkey's behavior, we ought to be able to find it with different random sets of neurons. It just looks different. So now I need my hands. I've got a three-dimensional hand, and if I hold it like this, it looks like one thing. If I hold it like this, it looks different, but of course it's the same thing. So they used a method called canonical correlation analysis, which essentially rotates your hands back and forth until they look alike. So they might, start like this, but if you rotate them both correctly, you can end up, well, I guess I have to do this. You can end up getting them to look correct. So they applied that to this 10-dimensional set of signals. Here are the rotated versions of them. And here's back where I was a moment ago. This is the decoding that they got if they made a decoder based on these latent signals instead of single neurons and did this alignment process on the data from day 45. Now they can use a single fixed decoder looking at these latent signals. So I will submit to you that these latent signals really are a reliable representation of the monkey's motor intent that is sustained for a long time, at least for these rather stereotypical, well-learned behaviors.
How do you think that relates to the changing neural signal versus like what you're recording? You're still recording units, right? So are they different neurons that sit within that same space?
The assumption is they are different neurons, yes. The possibility is a single neuron has changed what it wants to do on any given day. It's pretty difficult to answer that question, but I showed you those action potential shapes. We know, we know the action potential shape is changing. We can be pretty certain, at least in some cases, that it is a different neuron. It's harder to say conclusively that you really are recording the same neuron for an extended period of time because different neurons have similar looking action potentials.
So is the idea that this manifold is essentially telling you something about the connectivity within those series?
So there's a really interesting question. Where does this manifold come from? My money is on the fact that it's got to have something to do with synapses. There's a really nice story from Aaron Batista's lab at University of Pittsburgh that came out also about 10 years ago, in which they did a BCI essentially like this, based on latent signals rather than single neurons. They did two things to the mapping of that set of latent signals. One allowed the covariance pattern of neurons to stay the same. So basically the reason this happens is many neurons are doing the same thing. And so you can lump them all together and you find these latent signals. So one of their perturbations allowed the monkey to keep all those covariance patterns the same. The monkey could easily learn that kind of a remap within a single session. Another one forced them to modulate their neurons such that it broke these covariance patterns. Those were very difficult. No monkey learned one of these within a single session. It took Emily Obi careful training for weeks of small perturbations to get the monkey to do one of these out-of-manifold perturbations. So it's suggestive that something is happening to the real connections in the cortex in order to make that new manifold. I think it's very fascinating and we don't really know yet.
So you can look at these manifolds across different time characteristics then? I mean, if these are connectivity issues, I would think, from a data standpoint, there's a connectivity sort of piece, or there's a time correlation between connectivity of something.
You lost me there.
Well, I mean, if these are patterns of neurons that are firing based on, other network behavior, right? I mean, they're not going to be, they're all always maybe they are always simultaneous, but the network activity is synapses between them, right? So you presented sort of what I'm interpreting as a time snap, but I'm wondering if there's a correlation between prior activity and sort of progression through maybe different manifolds that you're capturing a time snap.
Well, so the implication of those results that I just told you about that span a couple of weeks, I agree entirely with you. The problematic part is when the monkey changes his behavior moment to moment.
So I was thinking time like microseconds, milliseconds, like pattern activity as it progresses through that portable region.
Can we talk about this at supper?
Dinner's a good time.
Dinner?
Yeah.
I need to think about this. I'll just say dumb things if I try to answer now.
Fair enough. I'll make the dinner better.
Yeah.
How much data is required on the new day to perform this alignment?
Not much. I'm not going to be able to get into this. In addition to this linear method, we looked at some GANs-based methods. Those took 30 seconds of data. So this is one of the reasons this might be another reason this might be advantageous. It takes us 10 minutes of data to make a decoder in the first place, but we can align two decoders with pretty little data. Good question. Another limitation that I need to make sure I don't forget to mention, this canonical correlations literally does time correlations between these 10-dimensional sets of latent signals. So you need to start by getting the monkey doing the same thing twice, because if the kinematics don't line up, there's no way the latent signals are going to line up. That's a fundamental limitation, one of the things we're working on now, but is not in this talk, but happy to talk about it at some point. Any other questions? Yeah.
Do you find out the monkeys do the same thing over and over again?
Yes, because we force them to.
Do you think that you grab something off the counter the same way every time?
Well, I mean, ask me that question in about 10 minutes. That's an important question. You certainly can do it nearly as reliably as the monkeys if you really want to, but you need to be motivated, and the monkeys are motivated. If you just grab something, you're not particularly, and there will be more variation. And another important question is, if you do it and I do it, are we going to do it the same way? Any other questions? All right. Oh, it works for 2 years as well as a month. It's really kind of amazing. All right. So in fact, now I'm beginning to get to your question. Now I'm making a subtle switch from monkeys making arm movements to monkey making isometric wrist torques. It doesn't really matter, but there it goes. So he's doing the same kind of center out behavior here. This is the same kind of neural state space representation of these 8 directions of force application. Looks more or less the same. This is a different monkey doing the same thing. Now this doesn't mean his force trajectories are different. In fact, they're rather similar, or this method wouldn't have worked either. But the latent space looks completely different, just the way it did when it's separated in time from the same monkey. So the obvious question is, well, can we use these same methods and rotate monkey S latent space back to monkey J, if I got those right? And the answer, not surprisingly, is yes, you can. So they use the same canonical correlation, got pretty good overlap between these two monkeys' latent spaces. So now they computed a decoder from monkey J and used it on monkey S. And this should sound like something that might be useful for a human. So here are those results. Black again is actual EMG here that we're predicting from a bunch of different muscles. And you see the R squared written on the right-hand side. Purple is within session, so it's as good as we can do in terms of predictions. Green is using the cross monkey decoder. Not quite as good, but pretty awful good. Using a decoder made from one monkey and applying it to a different monkey after aligning these latent signals. Yeah.
Does the similarity of alignment also correspond to, I don't know if you collected this data, the alignment of EMG activity to produce the same kind of task? I mean, so my thinking naively is if I'm going to do a task, essentially there's some similarity in the musculature to each individual because there's a lot of similarity in anatomy. And I'm just kind of curious if the muscle EMG has the same kind of ability to align from monkey to monkey as what your manifold shifts.
Almost certainly. We haven't done that, but it seems it has to. Those are more stereotyped than neural firing rates. So yes, you can, I mean, muscle synergies are basically that.
So that's kind of the base of the question because
Yeah.
this then relates to essentially.
Yeah, we've also asked questions, we have not asked that question explicitly, but we certainly have looked at dimensionality of EMG, see how that says anything, whether that says anything about motor cortical and premotor dimensionality. Emily?
How does the across monkey use of the decoder compare to within monkey across session?
If you're talking about aligned across sessions, it's going to be basically the same, a little bit worse. I'm sure they made that comparison. I don't remember exactly what it is. But if you look at these numbers here, they're pretty similar. And the proviso here is this is a simple, highly trained behavior that the monkeys are doing. And we need to have that in order to do canonical correlations. Any others? Okay. I already gave you the clue here. What happens if you, Emily? I put my picture up and then you stopped me.
I was raising my hand, you turned around. I actually forgot. Oh, man. Okay, I'll come back.
We
I'm
made fun of
sorry.
Bob and
I
he
was-
forgot things.
I was distracted. Sorry.
Okay, I'm sure it'll come back to you. So, the obvious question is, if we can ask a person with tetraplegia to try to do the same thing, does that work? And the alignment certainly did something useful. It doesn't look nearly as much as alike as the other monkey did, and I'm happy to discuss why that may be the case. I'm sure you can imagine a number of reasons why it would be the case. Nonetheless, again, the green curve's not quite as good, but pretty good predictions that we can make using a monkey decoder on data recorded from a human motor cortex. Okay, so this, in principle, gives us a way into doing EMG decoding in humans. If we can first make a decoder of the same behavior using this method in a monkey.
How did you do that? I mean, you don't have EMG. How did you do the human?
We said, try to make these force movements.
Okay.
That's it. So we asked him to do exactly what the monkey was doing. And we made the assumption that, back to your question, that the muscles are sufficiently similar that when he tried to do that, there would be a sensible pattern of activation in his muscle. I mean, really, we're making predictions of monkey muscles. And we know the anatomy is similar, so it ought to work fairly well.
But these were imagined human.
They're imagined, attempted, whatever you want to call it. Yeah, yeah, exactly. So it's the equivalent of the observation-based decoder in some sense.
What if somebody hadn't learned how to do this with FES?
Sorry, if someone what?
Yeah, we'll talk about it.
Okay.
Are these electrodes in the same place?
These electrodes are both in the hand area of motor cortex.
I see.
That's
In
as
both the species.
close as we can get. Yeah, yeah. So we're able to find a similar kind of, the assumption is we're able to find a similar kind of latent space in those neurons. Dustin, you look really unhappy.
I'm like, it's just, there's an awful lot of leaps here. I'm trying to keep.
But be nimble, Dustin. Emily?
With you, I'm skeptical.
I would think less of you if you were not.
Thank you.
I got my question back, but it's not as probably relevant. I was wondering if, the monkeys you were comparing across, if they had really similar like strengths and like degree of muscle activity. Like I'm thinking if you had one really bulky monkey and one really skinny monkey, if they'd be the same in terms of their
I don't think
activity.
strength is the issue, because there are lots of ways we can scale this thing easily. But different musculature for sure could be important. I mentioned muscle synergies. I can do the same thing several different ways by activating different sets of muscles. Maybe a monkey wanted to do it differently than Scott wanted to do it. Now, that's probably okay, because the way the monkey did it is a biomechanically feasible way of doing it. Scott may not want to do it that way, but it'll work. So there are lots of issues like these that, at the very least, we'd have to wrestle with. Anything else?
I just want to ask a methods question.
This is your last one.
No. That's a lot of pressure for the last question. Is
Just
this
don't forget it.
this is so is he moving wrist? I'm looking at the targets. He's center and then moving wrist, or what is the task
It
specifically
is
you're
an
decoding?
isometric wrist torque, so he's not actually moving, but he's exerting torques.
Yeah, okay. So it's mostly wrist musculature
Yes.
and hasn't okay.
Absolutely, and this, in fact, is going to come up on a slide in 1 second.
Then it doesn't count as a question if I was just prepping you.
Oh, great, you're good at it. Oh, shoot, didn't come up. This is just to point out, if Scott, who is the human participant, imagines making single finger movements, those also show up in different parts of the state space. So this is my segue by saying, even, we want to go beyond these very simple wrist movements, the geometry of which is really simple, to something more interesting. And at least within the brain, there is the prospect for doing that. Here is the simple muscle picture. For those of you who don't know, there are 4 muscles that do all this torque. And it really is a simple problem compared to what I want to do.
I was wondering if you go back to the last slide, if you do compound movements where maybe you're doing multiple fingers, does it show up again in like another, I guess, part of this?
If you want me to guess, I'm willing to guess. If you want data, not so much.
Okay.
In fact, I'm in the middle of writing a grant with one of the postdocs at the University of Chicago who's interested in not just that question, but even single joint version of that question. Almost certainly it's going to be nonlinear, and it's going to show up somewhere else. Anything else? Okay. Really simple muscles, really complicated muscles. That's the problem. So it would be feasible to do the kind of decoder that I just told you about if all we wanted the human to do was to move their wrist. If we really wanted to make this kind of movement, and if we're stuck using canonical correlations, it's hopeless. Because this means we'd have to have them doing the same thing with their hand that the monkey's doing with its hand. It's not going to work. We're working on ways in which we don't have to align the kinematics, but those are not working yet. This isn't working either, but at least we're setting out on it. So the plan now is to get a bunch of data from neurologically intact humans as they grasp a bunch of different objects. A bunch of data means EMGs, contact forces, and motion tracking. So we're going to know, everything about the hand that there is to know as you're grasping objects. We're going to take that information, we're going to ask the persons with spinal cord injury to try to grasp the same object. I think I'm getting ahead of not getting ahead of myself. So this is what I just told you, record all of this data. What I didn't tell you is we're going to go through a stage called computed muscle control. This is a bit of a detail. I may just gloss over it. In fact, I'm going to gloss over it. Basically, it's built around a musculoskeletal model of the hand from which we can infer the activity of muscles we haven't actually recorded. So now if I can record 12 muscles, forearm and hand from humans, I can, and if I have a good enough model, I can infer the activity of all the rest of the muscles. So now I have predictions of every last muscle in your forearm and your hand, in theory. And this is work with Wendy Murray, who has such a model. Okay. Then the next obvious step is we record M1 from our participants as they're trying to do the same behavior. Implementation. Record behavior, record from the participants. This is implementation of the decoder. Decoder training, this is, who put this figure together? We're going to record a decoder between the recorded M1 activity from the paralyzed participants and the recorded EMG activity from the intact participants. That gives us the decoders. Then we'll record from the participants, go through the decoders, go through the forward musculoskeletal model. That allows us to predict patterns of joint torques, of motion, of interaction forces, and so forth by using Majoko, which is a physics engine in a virtual reality system. So the goal then is initially to actuate this biomimetic version of the hand in virtual reality to grasp and squeeze on objects and so forth, and then implement it in a robotic hand. And basically go head to head between that kind of grasp control and the kinematic kind of grasp control that's currently being done. Which is not where I want to end this talk. Where I want to end this talk is the advertisement. This is just more of what I've just said. I'm sorry for those of you who are not used to looking at pictures of surgery, but this is what it looks like to implant a whole bunch of intramuscular electrodes in a monkey. Wires going all over the place. In the forearm, it's not that hard to implant a bunch of different electrodes. In the thenar complex, it's a whole lot more difficult. So the obvious solution is to use fine cuffs instead. So I've actually been collaborating with Dustin for quite a while to use the cuffs that he and Dominique have developed for many years. And I'm going to assume that most people are familiar enough with them that I don't have to belabor the issue here. But basically, they're multiple contacts that surround the circumference of the nerve after it's been gently reshaped such that the internal pressure does not go up very much, but the electrodes become much closer to the axons. Now, the benefit here is you get to hear Dustin dictating in the background.
On, second finger on, first finger off.
Oops, so much for the audio. I hope the audio works in the next one. So basically, we had control of the fourth and fifth digits from discrete different contexts in the fine. Yeah.
Where did you place the cup?
Ulnar nerve. In the forearm. I think it was below the elbow. Do you remember?
That was below elbow.
But that's a good question. And hopefully, Dustin is really proud of this.
This will be lateral connection.
That's Emily Obie for any of those who know her.
Do I have that back down, Leslie? Thank you.
It's kind of irreverent, but sort of cute. So I don't think he's going to be doing.
Morning, I believe.
Yeah, that's right. Dustin's job was to hold the video camera and to show up with a 10-pound bag of peanut M&Ms. That's what we needed him for. Katie, okay. So there is the potential for using these fine cuffs even for finger control, and you guys may well have better examples of that. But the big problem is I just had a conversation with Dominique, wherever Dominique is, right there, who reinforced that it's even harder than he ever thought to record from motor fibers in these nerves with cuffs. So we can't get a motor signal even from a monkey from which we might make a decoder. So we have to do something else. So Ben has spent a lot of time recording the responses in single muscles from different contexts across cuffs. And you can see, in this case, stimulating an axillary cuff, he gets a pretty nice independent activation of the posterior deltoid before the other two heads of the deltoid come on. And similar, more distal in the median nerve here. There is the potential for selective recruitment of these different neurons, and you can get these recruitment curves for all muscles and all contacts, but it's really slow and tedious. This is just single contacts. Now think about the interaction of pairs of contacts, let alone triplets or quadruplets. How on earth are you going to learn how to, the control space to activate the cuff in order to produce the EMG that my decoders say you need? So then later work with Flood is an attempt to simulate this system. I'm going to go very quickly through this, partially because I'm running out of time, and also because he's the one that should really be talking about it. But basically, what it allows him to do is figure out the field strength that results from stimulation of different contacts, and then the probability of activation of axons within different muscles, within different fascicles that go to different muscles. And if we can simulate all this, then Nikolai can make these activation surfaces here for two contacts to figure out how they activate muscles, and potentially for a higher order combinations of contacts as well. And if we can understand this mapping from multiple contacts, and even ideally, a nerve, imaged nerve of a human patient, so we know what their nerve actually looks like, then conceivably we can begin to develop models in silico of the activation from contacts to muscle. So that is, in fact, the last slide. I won't bother going through the summary. Emily.
So I've asked some of the students about this as well, but I'll ask you, how realistic are those fascicle shapes, and what role do you think that could play in this results?
So those fascicles came direct from real images, I believe, right?
Yeah, so they came from intraoperative ultrasounds on subjects we were with. And the one thing to note is that there's a chance that many of the clumps you see in the ultrasound are individual fascicles, but instead they're the compilation of fascicles, I guess. So maybe you don't want to use in future models more histological slices representing. They're from Ultrasound.
Yeah, I guess I'm just asking because they seem like they have like really sharp kind of corners to them, some of the fascicle shapes, and if that would potentially change the way the fields were shaped in like the FEM.
Yeah, so there's, yeah, I could see that, that might affect it. I could see that, that just has to do with the way that it was traced from the image, so that could definitely be correct.
So let me show the next 2 slides real fast, and then I'll be happy to take any remaining questions. And that's just the big list of lots of people, and it's by no means exhaustive, that were involved in these experiments and funding sources. And also this cortical bionics research group that has been formalized in the last half year or so. And also just have to point out, Sleeman Benzmea on the left was really one of the real driving forces for this consortium and is no longer with us as of the last few months. So, thank you.
How dependent do you think the range of possible in-one activation patterns is on proprioceptive feedback? And to what extent is this going to constrain your ability to generalize decoding methods to SEI patients?
Good question. I don't think that's going to be the biggest of our problem. It turns out FES is hard, and I don't have to tell you folks that. And figuring out how to stimulate the context is going to be hard. I think if, 15%, who knows? But I think my guess is that error is going to be smaller than other sources of error, but it's a guess. And it's something I'm very interested in. The other possibility, in fact, this cortical bionics group just had a Nature Com or Nature something paper a week ago talking about the activation of M1 when you stimulate S1 and its characteristics. And it turns out to be state dependent. So there is no single one-size-fits-all projection from M1 to S1. It depends on what the patient is doing. But our goal, of course, is to restore touch and proprioception by stimulating some of the sensory cortex, and in that case, reactivate M1 and put it back the way it should be. That's never going to happen the way I just said it. But it's all part of the same question, for sure.
Question along the same lines, since you brought it up, is
I've always been curious. Question along the same lines, not you bring it up, and we talk about this internally here, is the circuits engaged, for example, and this is more in the context of prosthetics, right? If I'm stimulating from the periphery, how much of the precortical circuit engages that loop, if you will, right? And so when I'm controlling movement, it's not starting in the perceptual part of the cortex, it's starting in the periphery and coming through, running through this through, all the other circuitry on its way up, right? Once it goes to the cuneate nucleus, it's all over the place. So what's your thought about interacting in the middle of that system and its ability to connect that control system and reconnect that circuitry versus, say, coming in from a
And
lower?
by starting in the middle, you mean by doing ICMS in the primary somatosensory cortex?
That is what I mean.
I sense that might.
Well, I mean, so that's a biased view, obviously,
Yeah.
but I'm just kind of curious about the periphery where, I mean, the basic question is the periphery versus engaging the entire circuit system versus somewhere else.
Well, I'm not even going to argue with you. If we could stimulate in the periphery, sure, it makes much better sense. Spinal cord injury, you can't. And as you know, it's tough to do that as well.
Well, in the spinal cord injury, for sure, but I know you'd worked in the cuneate nucleus for a little
Yeah.
while and tried to get at that, which is obviously surgically
Yeah.
challenging. But just your thoughts on if you could move somewhere else down
No,
the
I
circuit.
agree. Lower is probably better. Cuneate, for those of you who don't know it, is literally at the southmost tip of the brainstem, dorsal brainstem. And we had an experiment with Sleeman for 5 years. And we implanted 6 or 7 monkeys and basically recorded the properties of the cuneate. It would have been nice to stimulate the cuneate and ask the monkey what it felt like. We never got around to doing something like that. But that was part of the goal, in addition to learning something about the cuneate, to figure out whether it might be a more peripheral entry point for these signals. The cuneate in particular is pretty close to other pretty important stuff in the brainstem, so you have to be pretty confident you know what you're doing.
With the mapping of the monkey decoder to the human, have you validated in able-bodied, for instance, that the EMG from humans doing a similar task is similar? And if not, do you think that's where some of the error could have come from?
No, because the decoder didn't. As I admitted at one point, these really are monkey EMG predictions based on human M1. So the concern would be if the human's muscles really were substantially different from the monkeys, then it might not have worked. But we know from anatomy they're basically the same. There are differences between the musculature, but they're pretty minimal. Again, well under the error level we're going to have from other places. The bigger issues are with the method that I showed you. Scott had to be doing exactly the same thing as the monkey was doing, and to the extent they differed, the predictions are not going to be as good. And that's part of the reason for sure why that alignment of the state space didn't work nearly as well, because the timing of his activity and exactly the straightness of the torque projections and all that were undoubtedly not as crisp as the monkeys.
Thanks.
Yeah.
I have a question about the sort of lines in the aligned neural space for the monkey's behavior. So if the monkey do you think it would be the case if the monkey were to begin to do one of those tasks like move? Do you think in the aligned neural space, if the monkey began to do a task like move the wrist up and then deviated from it, that you would see it go along the line to the halfway point and then do something else? Or do you think because the activity is different overall, that it would lie on a totally different meaningful plane or whatever?
There's pretty good evidence that corrective movements, in fact, are in a different part of the space than the main movement. So the answer probably is, yep, that's a problem as well. How much we can get a decoder that doesn't care about these details and manages to do a pretty decent job of them all, I'm not sure. I have a former graduate student who's now faculty at the University of Montreal, and he has a collaboration there with Mila, the machine learning group of Yasho Benjios, and you may have seen Yasho is accredited up here as well as a consultant. And they're trying to produce basically the equivalent of a large language model network trained on a gazillion seconds of not just monkey data, but monkey data, rat data, whatever we can give them, hoping that it becomes sort of the universal Babelfish translator for all these things. So who knows? But it's the kind of project that takes $10 million of computation time, let alone anything else. So it's way beyond anything I could do. All right, thank you.